Hey everyone, welcome back to another TEAS review lesson. Today we're going to look at chromosomes, genes, and DNA. These are the objectives from the ATI study menu. You are expected to understand the function of chromosomes, genes, and DNA. Also be able to explain the relationship between chromosomes and genes, and also differentiate among the structures of chromosome, genes, and DNA. Okay, let's first look at the structure of DNA. So the DNA is this double helix, right? That really looks like a ladder, a twisted ladder. As you can see, this is a one strand, and this is another strand. The two strands are held together by hydrogen bonds, right? These dotted lines all represent hydrogen bonds. You can see the hydrogen bond is formed between bases, right? Bases from two nucleotides on the opposite strands. Now, these polynucleotide strands, so polynucleotide really just refers to each DNA strand having multiple nucleotides, right? DNA is a polymer, and the monomer is nucleotide, right? This is one nucleotide, this is another nucleotide. So you can see there are multiple nucleotides in each strand. So the two DNA strands go anti-parallel. That means that these two strands go opposite directions. Believe it or not, each DNA strand does have direction. As you can see, we indicate direction by 5' and 3'. Now, I don't think you need to know why this is 5' and why this is 3'. Just remember that one strand goes from 5 to 3, and the other is going to go the opposite direction, right, which is 3 to 5. So you can see the directions are totally opposite. Now, these two DNA strands are complementary following the base pairing. You need to remember this. A pairs with T and C pairs with G. This is, some of the most basic information you need to know about DNA. So this base pairing provides a perfect mechanism for DNA replication and also for the information flow from DNA to messenger RNA. And now we know that the genetic information is stored in the sequence of nucleotides, right? So there you go. My pen stopped working for a second. Now, because of the 4 bases, it generates 4 different kinds of nucleotides, right? So these nucleotides can be in so many different possible orders, and this leads to the genetic information, right? Basically, the DNA sequence or the gene sequence. Okay, now when we want to use the information from a DNA, basically from a gene, to make a protein, we have to follow 2 steps. The first step is transcription. You are copying or transcribing the information from a DNA to messenger RNA. And usually this is on a gene basis, right? So each gene will be copied to the prospective messenger RNA for that gene. The second step, which is called translation, is going to use the genetic information to direct protein synthesis, right? And this is done by ribosomes. Now, ribosomes are located in the cytoplasm, but DNA is located in the nucleus. It cannot get out of the nucleus. So you need a little messenger to relay the information from DNA to ribosomes so that ribosomes can use that information to make proteins. We are going to use messenger RNA to deliver that information because mRNA can get out of the nucleus, right? Because mRNA molecules are very small. They can go through the nuclear pores and access the nucleus and deliver the information to ribosomes. Now let's look at transcription first. So in transcription, we are using only one of the 2 DNA strands as a template strand to synthesize messenger RNA. Now the mRNA is going to be synthesized using the 3' to 5' DNA strand. So you can see this one goes from 3 to 5, right? So we're going to use this as a template strand to make mRNA. Now, when you make mRNA, you have to follow the base pairing rules, right? So that you can copy the exact genetic information. So if this is an A, then the mRNA, which goes from 5 to 3, because the 2 strands have to go anti-parallel, whether it's 2 DNA strands or 1 DNA, 1 RNA strand. So I'm going to use a different color to indicate mRNA. The first mRNA nucleotide is going to pair up with the A, the adenine nucleotide in DNA, right? But remember, in mRNA, there's no T, right? In RNA molecules, there's no T. So instead, A is going to pair with U. So the first nucleotide is going to be U. And then G pairs with a C, right? The second nucleotide in mRNA is going to be C. T pairs with A, and C pairs with G. So this is going to be the sequence in this particular segment of mRNA. All right. Now, over here, the information says that the mRNA sequence is going to be the same as the other strand. So if this is the template strand over here, this is the other strand that text was talking about, was referring to. And it is the same, right? If you look at the sequence for this other strand, it's T, G, A, G, right? And when you go back to mRNA, it's U, C, A, G. So remember in RNA, there's no T, so instead you have U. But you can think of U as the T in RNA molecules. So the sequence is exactly the same, right? So that's why we call the template strand, the strand that you are using to make mRNA, the antisense strand, meaning it's not the coding strand that we use to synthesize proteins. But the other strand, this is the sense strand, meaning that its sequence is exactly the same as the mRNA sequence, and this particular sequence is going to be used by ribosomes to make proteins. Okay, so that's what sense strand and antisense strand mean. So again, the sense strand is going to be the coding strand, right? Because the sequence of the sense strand, that's exactly the same as the mRNA, and that's the sequence used to make proteins. Now, normally, I don't see this information in TEAS study manual, but in this latest version, they did mention the sense strand and the antisense strand. So I decided to put the information in here just in case. Okay, now let's look at the second step in gene expression, which is translation. So now you have your little mRNA sequence, right? And now we're going to have ribosomes coming in and use the sequence information in mRNA to make proteins. Now, how do ribosomes read the genetic information in mRNA? So how do they know, which amino acid to grab, right, to add to the polypeptide chain? Now, the ribosomes are going to follow this particular rule. But first, we need to talk about what codons are. So codons are 3 nucleotides collectively that code for 1 amino acid. So for example, UCU is a codon that codes for serine. So this is a codon table. And this is how you read the codon table. The first letter is on the left side, right? So you start from the left side. So you're going to find U first. So this is the first letter. And the second letter is on the top. So the second letter is C. So you find C next. And you will know that this amino acid that the codon codes for is going to be in this box. But there are 4 codons, right? So you need to find the third letter, which is U. So it corresponds with the first codon. So UCU codes for amino acid serine. So that's the rule that ribosomes are going to follow. So ribosomes are going to read the mRNA sequence in sets of 3, right? And particularly sets of 3 nucleotides. When a ribosome reads UCU, then it's going to add a serine to the growing polypeptide chain. Now, when you look at the table, there is a start codon, which is AUG, which codes for methionine. So the start codon is where the ribosome is going to start the translation, right? If you see mRNA sequence here, So maybe you have UCU, AUG, CCC, right? And then you have UGA, CCU. The ribosome is going to scan through this sequence, and it only starts translation when it gets to the start codon, which is AUG, right? So when it gets to AUG, it knows, okay, I'm gonna start grabbing amino acid, and then I'm gonna make a polypeptide chain. So it's going to add a methionine, and then it keeps iterating, right? Now it's gonna go to the next codon, which is CCC. So C, the second column, and then C here. So CCC codes for proline. Okay. And the next codon is UGA. So UGA is right here. So this is a stop codon. So when the ribosome gets to the stop codon, it's going to stop translation. It doesn't matter if you still have codons after UGA. It doesn't matter. When the ribosome gets to a stop codon, it is going to stop translation. So this is going to be the amino acid sequence of the polypeptide chain. So you can see the sequence has methionine and proline, but of course it's really short, right? Because we're trying to save time. I don't have the entire sequence here, it's just for demonstration purposes. So now you know how ribosomes read the mRNA sequence and synthesize proteins. Okay, now let's look at gene. So you know what DNA looks like, right? A double helix, and it can be a very, very long string. And in fact, the DNA is about 2 meters long, and you compile the DNA into a tiny little nucleus in a tiny little cell, right? So you can see there is a lot of packaging there. Like I said, the DNA can be really, really long, right? And in this DNA string, there are segments that code for particular proteins. Okay? So for example, if we look at this DNA string, this section codes for a protein. So that's a gene, right? That's basically what the definition is, segment of DNA that codes for a protein. And then maybe there's another segment downstream, and this codes for another protein, so that will be another gene. Okay? So parts of the DNA that codes for proteins, they're called genes. Now, some genes do code for RNA, so not all genes code for proteins. So this gene could be the hemoglobin gene, right? And this codes for hemoglobin. I'm just going to do HB, hemoglobin protein. Now, chromosomes are really kind of at a larger scale than the DNA and the gene. So when you look at DNA, right, the DNA is a very, very long string. If you don't do anything, you just put a DNA string in the cell, it's going to tangle, right? So think about the threads in your sewing kit. The threads are very easy to tangle. And this makes DNA very susceptible to fragmentation, right? It can be damaged very easily. So here is the mechanism to kind of protect DNA, where the DNA, like a thread, right, wraps around some proteins like this. Okay? These protein complexes are called histone proteins. Histone proteins. Now the histone proteins are like a spool where DNA can wrap around, and this kind of keeps DNA organized so it doesn't get tangled, it doesn't get damaged. And this is going to form some subunits, and I call these subunits nucleosomes. So you can see each nucleosome is a histone protein complex with some DNA wrapping around it. Okay, so this is one nucleosome. And then you're going to have multiple nucleosomes because you need a lot of packaging, right, to fit the very long DNA in a tiny little space of the nucleus. And just imagine, thousands of nucleosomes together that form chromatins. So chromatins are really just a combination of DNA wrapped around histone proteins. Now, chromatins will condense during cell division. Now, normally chromatins are, kind of thread-like. You don't really see them. under the microscope. But during cell division, chromatins will condense. So there's a further compacting, further condensation. And this will form chromosomes. So after the chromatins are condensed, they are going to have this kind of typical X-shaped structure, which we call chromosomes, right? So a lot of people are familiar with the chromosomes, but they don't know that chromosomes only appear during cell division. Other than cell division, the chromosomes are kind of in this chromatin state, which are really thin, very hard to see. But once the chromatins condense, then they become chromosomes, which are more visible under the microscope. And this is what they usually show, right? The typical X shape. And that's because during the initial stage of cell division, DNA and the chromosome are duplicated, right? So now you have two identical chromosomes. So that's why you see this typical X shape. But really, there are two sister chromatins that are connected, which form this X shape. Now, prokaryotic organisms normally have a one circular chromosome. So it's a circular, and there's only one chromosome because there are very small organisms. Now, prokaryotic organisms are bacteria. So their genomes are pretty small. So usually they just have a one circular chromosome. For eukaryotic organisms, which are typically bigger, they have more genes. So they will have more chromosomes. And usually those chromosomes are linear. So you can see in the picture here, which demonstrates the human chromosomes. So this is a karyotype of human chromosomes. You can see these are linear, right? And then there are multiple chromosomes. We have identified the different chromosomes in human cells. So you can see we have numbered them. This is chromosome 1, chromosome 2, and all the way to chromosome 23, which are the sex chromosomes. So you often hear, DNA sequencing companies like 23andMe. So that 23 actually comes from the 23 unique chromosomes in human cells. We have 23 different kinds of chromosomes. You're going to think, why don't we have 23 chromosomes? Why do we have 46 chromosomes? That's because for each unique chromosome, for example, chromosome 1, you have 2 copies, right? So this is copy number 1, the copy number 2, because you have 2 parents, right? So one copy comes from the mother, the other copy comes from the father. So again, for each chromosome, each type of chromosome, you are going to have 2 copies. So 23 by 2, that's 46 in total. Cats have 19 pairs of chromosomes, right? So they have 19 unique chromosomes, and each chromosome has 2 copies. So that's a total of 38 chromosomes. Dogs have a little bit more chromosomes, the most among all the 3 species. So dogs have 39 pairs. Now, chromosome number is unique for each species. As you can see, humans, cats, dogs, we all have a different number of chromosomes. Now let's look at the relationship between DNA, genes, and chromosomes. I have seen questions with some statements on the relationship between the 3, and you're supposed to pick the correct statement. So again, DNA is this long string, right? And on DNA, there are segments. There are different, parts of the DNA that will code for proteins or RNAs. So these segments are genes. So basically, genes are part of DNA. And then DNA wraps around histone proteins, right, to form this complex to compact DNA and keep DNA from being damaged. So this complex of DNA and histone proteins are called chromatins. Now, chromatins will condense during cell division and become chromosomes. Okay, so again, chromosomes are really kind of on a higher level, much bigger, than DNA and genes. Right? So if you have to go from small to large, the smallest is going to be genes because there are only segments of DNA. And then the next level is going to be DNA. right? DNA can contain multiple genes. And then when you have a DNA and histone proteins, that's going to be the chromosome. Okay, so that's the relationship. All right, now let's look at some practice problems. Number one, which of the following base pairing is correct? You have to memorize this. A pairs with a T and C pairs with G, right? And in case of transcription, then A is going to pair with U. So the correct answer is which one? B, right? Number two, if the DNA of a particular species contains 15% C, how much T do you expect in the DNA? This is a question that I use a lot in my biology class. And this is designed to test your knowledge of base pairing. So C pairs with a G, right? So that means in the DNA molecule, the percentage of C should be equal to the percentage of G. So if C accounts for 15% of the DNA, then that means G is also 15%. All right. Now, in total, C and G together, that's 30%. Right. So that means the rest, which is 7%, right? 100% minus 30%, that's 70%. So the rest of the DNA is all going to be A's and T's. And you also know that A pairs with a T, so A and T should have the same percentage, which is half of the 70%. So A is going to be 35% and T is also 35%. So which one's the correct answer? Oh, actually, I didn't put the correct answer here. That's probably a typo. I think I was supposed to change that to 35%. That 30% comes from if I change C to 20%, then the correct answer would be 30%. I forgot to change it back. Okay, so correct answer is 35%. Number three, what do the majority of genes code for that determine traits? Genes code for proteins, right? And proteins will determine your trait. Number four, which of the following statements is correct? So this tests your knowledge about the relationship between DNA, genes, and chromosomes. Now let's look at these options one by one. A segment of DNA is chromosome. Now that's not right, right? Chromosome is made up of DNA and histone proteins. So chromosome is, a much bigger structure than DNA. Or the correct statement should be a segment of DNA that codes for a protein or RNA is a gene. B, chromosome is made up of DNA and histone proteins. That's the correct statement. C, genes are a segment of RNA. Now you have to be careful. I made this question to be a little bit tricky. So this should be DNA, right? It's not RNA. Genes are segments of DNA. Last one, each chromosome codes for a protein. Now that's not correct, right? Because each chromosome has the entire DNA. And there are multiple genes, many genes. It can be many genes on a particular DNA. So you're going to have multiple genes. So this will code for multiple proteins, right? You need a plural. It's not just one protein. All right, we're done with this lesson. Good job, guys. Keep studying until I see you next time.